So today we're going to talk about protein, and we're going to have some of the structure, classification. I need to go through and pull all these animations off because this just drives me nuts now. Back in the day when I had learned how to do it, I thought it was really cool. Drives me nuts. So we're first going to talk about structure, and I know we talked about this a little bit. So do you guys know the main difference between, or what additional thing protein has as far as an element on it compared to carbohydrates and lipids?
It has an amino group.
An amino group, what's the main element in the amino group? Nitrogen.
That sounds right.
So our protein structures, we have the basic structure of a protein is an amino acid, where you have this acid group, the COOH is the acid, and the NH2 is the amino group. So that's where we get the name amino acid. So within it, we have a center carbon and there's a hydrogen. And do you guys remember how many amino acids there are that make up protein?
Is it 27? It sounds
13.
No. Zach, you know. So there are 20 that make up protein. There are, within all biological functions, there's probably an additional 7, but they don't make up protein. So the thing that differs among all these amino acids is this thing called the R group. So the R group is the only thing that changes. The base structure is this acid, the COOH, in the amino group. Center carbon with the hydrogen, and then that R group. When we have two amino acids put together, that forms a peptide. So this would be a dipeptide, di meaning two, peptide meaning amino acid chain. And you can see here that we have the oxygen from here is double bonded to the carbon, and then we lose this OH, which is the hydroxyl group, leaves, and then we end up taking one of the hydrogens off of the amino group, so the nitrogen binds to the carbon. So if you had this hydroxyl group removed and this hydrogen group removed, what do you think the byproduct is of making a peptide?
Water.
Water. So we end up making a water for every 2 amino acids that we bring together. So all proteins have these common properties. When you get chains of these, they're called peptides. Now, our protein, is it just long chains of peptides? Is that how protein works? Just long chains of amino acids? No, so we end up getting different structures. So we have the primary structure, a secondary structure, a tertiary structure, and a quaternary structure. So our primary is the single peptide. Secondary is where you start having, you may have heard of alpha helices and beta pleated sheets. Have you heard of those?
I remember it from biology, but I don't remember anything about it.
So this, you end up getting more of a two-dimensional shape. So primary is this long. Once you get more than 20 amino acids, they're gonna start, those amino acids are gonna start interacting with each other. And those things that are interacting are gonna be these hydrogens. And you end up getting hydrogen bonds. Now, if you remember anything about your general chemistry, hydrogen bonds really don't cost any energy or anything to make. It's just kind of naturally occurs. You're gonna have, depending on these R groups, some of them are gonna have a positive charge, some are gonna have negative charges. So all of these are gonna end up interacting, and so there's no energy that goes into making these secondary structures. Then we have the tertiary, and the tertiary, it ends up having, we have some amino acids that have sulfur in them. So we end up having these things formed called disulfide bonds. So these are contributing to the tertiary.
Is that supposed to be a beta?
What?
It's alpha.
It's the Greek, it's the
Oh,
Greek
oh,
egg.
oh,
Sorry. So tertiary, you end up having hydrogen bonds and these disulfide bonds. And quaternary ends up where you have more than one protein come together. So a good example of a quaternary structure is going to be hemoglobin. So you have 4 different proteins that come together to make hemoglobin. Where do you find hemoglobin?
Blood.
In your blood, in your red blood cells. So you need to have all 4 of those come together for hemoglobin to work. So that's where, so all functional proteins are going to be tertiary. Only some of them are going to have quaternary structure. Does that make sense? You tracking, Zach?
I'm tracking.
Okay, we went over before, a few weeks ago, so we're going to have a little quiz. What are our essential amino acids? So we'll go, there's a total of 22 amino acids. 10 are essential. Do you minimum remember the abbreviation?
Oh, it's, I know what you're talking about,
Is
but
it E omegas, right? Like omega-3 and 6?
Yep,
That's
it's your
what-
fats.
Okay. Afterwards.
it's,
Think military.
yes. What?
Here, I'll help you out. Private Tim Hall.
It's, oh, here it is. It's, PHE, VAL, TRP, PHS, ILE, MET, ARG, HIS, LEU, and LYS.
So these are, this is why I like to do this repeating thing, because now you're more likely to remember it, right? So our essential amino acids, phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, and lysine. Now, do you guys remember when we were making our diets for class, which ones are going to be the most common you're going to see in a feed mill and incorporating into a diet?
Lysine?
Lysine, we have lysine, threonine is number 3. What's the second one? Second most common.
It's, methionine.
Methionine.
What's that one? Sounds the most familiar, so I'm gonna go on over.
So these are what we call our limiting amino acids. And that in the diets, the feed ingredients that we're providing, these 3 are typically in the lowest concentration. So we could overfeed soybean meal to meet these needs, or you can add in a synthetic amino acid. So that's why we add these in separately as feed ingredients.
So is it like dietary supplements, like you can buy it like in GNC store, we just use that and put it in animal food?
Basically, so you can think of it that way. But the benefit of us adding them into the diet for our animal feed is it causes us to actually reduce the amount of crude protein that we put in the diet. So, because if you have to feed enough soybean meal to meet your lysine requirement, you're gonna be overfeeding all those other amino acids. What does the animal have to do to all those other amino acids that are above their
Break
requirement?
it down, place it in
What's the byproduct of breaking down the amino acids?
water. But,
More toxic than water.
Carbon dioxide?
They do make carbon dioxide. But what's the thing that you walk into a house and it cause your eyes to burn?
Ammonia.
Ammonia. So you're gonna get the uric acid production in the ammonia. So that's essentially what's leading to what we would call hot manure. And so they're just pooping all of that out. So it's costing you money because you're overfeeding a nutrient that they don't need, and it's causing potential pollution issues. So by feeding those limiting amino acids, we can reduce pollution and reduce the cost. So if you look at protein classifications, we have very simple proteins. So we have those that are yielding only amino acids. Albumin is the most common protein that you're going to find in your blood. It's soluble in water. It carries a lot of your other nutrients. A lot of your minerals, vitamins are going to be bound to albumin, and that's the way to keep it from reacting with other things within your blood. And then we have globulins. So these are you can get them to precipitate out. So your globulins are going to be like your immunoglobulins. We have elastins. So these are fibrous proteins. So this makes up about 30% of your total protein in the body is making your connective tissue. So this is your collagen and your elastin. So where have you heard about collagen before?
Muscle rebuilding?
So in your muscles. So why do you guys like to cook?
I do.
If you get a chuck roast, what's the best way to cook a chuck roast?
Smoke it.
Crock pot. So what's the crock pot doing? It's a slow cooker. So you end up melting all the collagen, and that's what's making all the meat fall apart. So the collagen is that connective tissue protein holding everything together. So collagen's insoluble in water. you convert it to gelatin and you can digest it. Elastins are kind of similar. Elastins are the things that as you age, you end up losing. So elastin's what's keeping wrinkles away, keeping things from sagging as you get older, everything kind of. So your elastins, that's why they call them elastins. They hold a lot of things in the proper place. And then we have what we call our conjugated proteins. So these are ones that are bound to the other nutrients. So we have glycoproteins. So those are proteins bound to a carbohydrate. We have phosphoproteins. So those contain phosphorus. A lot of our phosphoproteins are enzymes. So adding a phosphorus to an enzyme is going to either activate it or deactivate it, depending on the enzyme that you're talking about. And then your lipoproteins contain lipids. Where have you may have heard of lipoproteins? Medically.
Like plastic surgery,
Well, that's to suck out your adipose. So lipo means lipid. Have you guys heard of bad cholesterol and good cholesterol? So that's actually not cholesterol. What they're measuring is your lipoproteins. So your lipoproteins are proteins that are found on the LDL or ADL, not ADL, LDL, HDL, VLDL. And that's actually how we end up classifying the different lipoproteins. Do you know that?
Yeah, I got it.
So some terminology, we have true protein. And this is if you have a diet and you're giving all your protein is met with crystallized amino acids. So those are true proteins. They're nasty diets to make. The animal doesn't want to eat them. But that's how we've determined a lot of the actual requirements that an animal has. So you can decrease the amount. With non-protein nitrogen, so these are things that are contributing nitrogen but not protein, but you can convert the nitrogen source to protein using bacteria. We don't use this in poultry or fish, but ruminants do. Do you know what you feed ruminants, Zach? So they'll actually put in urea into a ruminant diet. So ruminant, they have all their fermentation happen before their small intestine. So those bacteria can take that urea and convert it to amino acids and microbial protein, and that's actually the protein that the animal is actually breaking down. Then we have what's called digestible protein. So this is the portion of the crude protein in which the animal can digest. So for our study that we're doing for class, one of the things we're determining is how digestible that insect meal is. Right, so that's supposed to be a protein source. So not everything is 100% digestible. So we're going to look at, well, I guess since we've talked about it before, how would you determine digestible protein?
Is that using the calorimeter bomb?
You're not using the bomb calorimeter, but It's a different assay, but the principle's still gonna be similar.
That's where you go all the way down to the net digestibility, or gross digestibility.
So we're gonna start off with gross digestibility, right? So we would have our protein source. And how would we determine what's digestible off of it? Similar like if we were going to get digestible energy. What do we have to account for? So there's no really good way to measure what they're breaking down, but we gotta look at what goes in and
What goes out?
what goes out. So what's coming out?
All the stuff they didn't need.
All the stuff they didn't eat, which is the,
So, like uric acid and stuff, is that what you're asking?
yeah, where's that gonna go in a bird? Yeah,
Yeah.
so it's poop.
Okay.
So we're looking
yeah.
So we're gonna measure what goes in and what goes out. What comes out is the poop, so that's giving us the digestible protein.
So you just weather poop.
Well, you end up, so you collect the poop and you dry it down, and we do, instead of bomb calorimetry, you do Keldols, which is a method of measuring nitrogen, or LECOs. We have LECOs now. And then you have absorbable protein, or which also is known as metabolizable protein. So this accounts for the quality and the quantity of the protein, and is accounting for more of what the animal has utilized. So your metabolizable protein, so I can take a, so what do you think is gonna be more metabolizable to, let's say a chicken? Soybean meal or poultry byproduct?
Soybean? Because it's closer to what they normally eat, not what their body is.
Do you have any thoughts? What's more utilized, easier to be utilized? Soybean meal or poultry byproduct?
I would think that poultry byproduct.
So the poultry byproduct is more metabolizable because you don't have that plant cell wall and that, so it's easier to get down and break down all those amino acids, break them down and metabolize them more so than soybean meal.
So is it always going to be the animal byproducts are easier to break down because of the lack of cell wall?
Yeah, animal byproducts are always more digestible and metabolizable.
How come they don't feed more of those then?
Cost.
How,
And some consumers think that it's
wrong.
wrong.
So,
I don't know.
We'll do it anyway.
Because a lot of consumers think a lot of things are wrong.
So that's where you'll see, if you go, some of the marketing thing, you go to Harris Teeter and try and buy chicken, some will say, you know, all veg diet, so they've never been given any sort of animal product, along with no GMOs, no antibiotics ever. So now we have differences in the quality of protein. So things that we can measure to determine the quality, one thing is the biological value. So the biological value is, there, that's the full equation, is looking at the relationship of how long the protein is retained versus how much is absorbed. So therefore, you take how much they ate minus what was in their urine and the feces. The nice thing about poultry, these are combined, and then you subtract, divide it by the difference of that, multiply it by 100. So that's giving you your protein quality. Another method is the protein efficiency ratio. And this is not a good method. I'll start by saying that because this is making a lot of assumptions. So protein efficiency ratio is, you compare protein sources based off of how much weight the animal has gained. So you look at body weight gain divided by the amount of protein that they had consumed. What are some of the things that you can think of that would be affecting that? Sickness,
Water retention.
water. What about other deficiencies? The amount of salt that you have in the diet. All of those things. There are a lot of other components besides protein that's contributing to body weight gain. But those are some of the things that we look at.
So would you say that's not really reliable?
It's not very reliable. It's easy.
Yeah.
So if you have a head-to-head, like everything is the same except instead of soybean meal, you put in the insect meal, like complete replacement, you're more likely to see that, but sometimes you're going to have different things are going to change. So it makes it difficult to do a straight exchange. So this is looking at how we're breaking down our protein. And I know this is, I need to find a better, in my 415 class I got better pictures of this. I have actual diagrams. So we have our dietary protein comes in, and protein digestion starts in the stomach, the true stomach. So with our poultry is going to be the proventriculus. And this is where we have the hydrochloric acid and the pepsin. It starts breaking down those tertiary structures, quaternary structures, tertiary structures, and the hydrogen bonds. So the combination of these two things, we now get those long chain peptides, about 20 amino acids long. Those then get moved into the intestine. The first part is the duodenum. We're going to have our cholecystokinin signal our pancreas to release zymogens. Do you guys know what zymogens are? So these are enzymes that are used to break down protein. The reason why we call them zymogens is they are released in an inactive form. Why do you think it's critical for them to be in an inactive form in the pancreas? So they don't eat the pancreas and result in eating you. Because the protein, if you think of it, protein is the workhorse. It's the thing that is making all the cells run. So you can't break down carbohydrates to get ATP without protein. All of those steps, we're utilizing enzymes. And what are enzymes made out of? Protein.
Also, not too easy.
And yes, lipids making up the structure, but lipids, it does do some signaling and stuff like that, but the main thing is you gotta have protein for those signals. So protein is like the workhorse of everything. So if we don't have them in an inactive form, we're gonna just eat ourselves through. So once they come back to the intestine, so the main ones that we have are called trypsin, chymotrypsin, and we have carboxypeptidases. So these come into the intestine and they get activated in the intestine. So they're released as trypsinogen, chymotrypsinogen, and carboxypeptidases. So these get activated in the intestine, and then they're able to break the protein up. These carboxypeptidases, they are moving, so they're what we call an exopeptidase. meaning that it's working on the end of an amino acid. It's actually working on the carboxyl end, so the OH group, and works down and it cleaves individual amino acids off a peptide. Your trypsin and your chymotrypsin just goes into the middle of the peptide and breaks it up. Within our intestine, we don't have just our dietary protein. We have our protein that is secreted with it. So a lot of hormones, any of our enzymes that stop working, we'll just break them down and then we can reabsorb those amino acids. Any of our cells that die, we're going to be breaking those down also. And then that results in absorption of our amino acids and our peptides. Does that make sense? Okay. So what this is essentially going through the metabolism. So we have our dietary protein, it's digested, which was what we just showed. So we have our single amino acids. Those amino acids are going to be used to make body protein, and I actually have those switched. Those should be switched. So synthesis should be over here. So the amino acids is synthesizing body protein, and then our body protein is degraded. So this actually ends up being a little cycle. So as your protein, your enzymes get worn out, we're going to be breaking them down, utilizing those amino acids to make more of that. So that's the cycling. But we also will metabolize these for energy. So that's where we get our gluconeogenic amino acids. So we'll break them down. Our byproducts be ammonia, urea, and uric acid. So that's where the nitrogen is. We have to take that off. And then the carbon goes in and it's able to go into our TCA cycle, /Krebs cycle, /citric acid cycle, because all three of those refer to the same thing. And this is where we're getting our energy. What's the form of energy we're getting? ATP. So you get CO2 and energy. If you have a lot of these, you can have them end up making fatty acids. Some of these will go and make glucose, so that's our gluconeogenesis. Some of these amino acids are going and making non-protein derivatives. So these include some of our vitamins, like our tryptophan is used to make dopamine and serotonin. It's also used to make niacin. And so we're making other vitamins and we're making neurotransmitters off of them. Does that make sense? Does your head hurt? Okay. So we want to provide an ideal amino acid balance. So we want to make sure that what we're feeding to the animal is meeting its nutritional requirements without overfeeding them. So that's called the ideal amino acid balance. So we'll look at what the requirement is for optimal growth and production and maintenance. We don't want excesses and we don't want deficiencies. Why don't we want excesses? We get the ammonia, and what else does that do to the diet?
Makes it non-effective.
It's more expensive is the problem. So it costs more. Here's another question. Can you store protein? Yeah.
Well, I mean, you can for like all kinds of like enzymes.
So we can store carbohydrates, right? How do we store carbohydrates?
Really,
There's a storage form of carbohydrates. that you know if you exercise a lot. The whole reason for carb loading before race.
No, just follow up.
It's glycogen. So it can store glucose in the form of glycogen in our muscles and in our liver. So if we need to run from a bear, we have the energy to get us to run for that short amount of time to get away. So that carbohydrate is stored for our fight or flight response. We're either gonna have to flee or we're gonna have to fight. So that's what we have, that carbohydrate there. Can we store lipids? Yeah, so that's all of our, in our adipose, we store it as triglycerides. Can we store protein? So the only thing that we have that you can think of as a storage form is to get muscle. So muscle is it's the lowest on the totem pole for the amino acid requirement. So if we put in excesses, they're breaking them down to store glycogen, or they're converting them to fat. And if we have deficiencies, they're not going to grow efficiently. And we also, have our, express this as, the amino acid requirement is expressed as digestible amino acids. So we can split this up. We have apparent digestibility, or in those books when we were looking up the feed ingredients, it was total digestibility. You guys see the difference in some of them, especially the Ebonic book. We would say total versus ileal or metabolizable. So apparent is essentially what we talked about before. You look at what comes in and you look at what comes out, and you are making an assumption that anything that is different is because the animal absorbed it. Is that the best thing? No. So what are other things that we need to account for?
What they excrete.
So what they excrete, so you'll have the enzymes that they're making, that any of the enterocytes or whatnot, any of the cells that die and they get broken down. So you're still contributing a little bit of protein in the intestine, even if you're not eating something. In addition, the microbial digestion is one of the things that we are most concerned about. So microbes are the things that you just need nitrogen, and you need nitrogen and carbon, and it can make any amino acid. So you can feed something high in methionine, but if the bacteria don't need methionine, they're going to break it down and make glycine or whatever. So if you see a lot of glycine in the poop, you're going to think your glycine wasn't digestible, where it could have been that it was just methionine that the microbes changed to glycine, and then methionine is not digestible. Does that make sense? So that's where we get this ileal or digestible amino acids. I feel like I've lost you guys.
It's just right.
Where did I lose you, Zach?
A while back.
So you all are nutritionists, so what would be a way to differentiate? So we know a parent is looking at the whole thing, collecting the poop, right, and comparing to what we fed. So what would be, how would we calculate, as a nutritionist, how would you calculate the ileal or digestible amino acids?
You know how much if you put in it, that you just measure what came out and then subtract.
Well, that's giving you a parent.
Oh, oh,
So how would we account for or get around this microbial to the best of our ability? Where's the majority of the microbial digestion gonna happen?
Or no, small intestine. Right.
Where's the majority of the microbial gonna happen in a bird? There's two of them.
This is the bronchitis.
Nope, that's the stomach.
I can see it on the diagram.
Cloaca?
No, it begins with a C though.
Sorry, Zika.
Zika.
Oh,
So
that's different.
one way that we can account for this is that we can euthanize the bird and take the ileum. Which section of the small intestine is the ileum?
It's the middle, right?
The first part.
No, it's the bottom. It's either the middle or the small intestine.
It's part of the small intestine.
Yeah, it is part of the small intestine. Which part? There's 3 sections of the small intestine. What's the first part? Duodenum?
Jejunum?
Jejunum?
So the
Ileus. So the ileum's the last part, and there's the least amount of absorption happening in the ileum. So you can euthanize the animal and take what we call the ileal contents. So you then collect the feces before you euthanize them, and then you euthanize them and you take the ileum. And then you can compare apparent versus ileal.
Is that what we're doing with those birds? Right.
So Thursday, if you guys want to have the opportunity of extra credit. So on Wednesday, we're gonna weigh for class because we only got an hour, right? So there's not enough time to sample and all that other stuff. But Thursday at noon, we're gonna euthanize all those birds and take the ileal contents. So then we can, and on Monday, Tuesday, and Wednesday, we're collecting all the feces. And so Thursday, we're gonna get the ileal contents. So we can compare total digestibility versus ileal digestibility. Does that make sense?
What birds?
It's quick, I promise. What's another way? So is there a way that you can get ileal that you can think of without having to euthanize the bird?
Is that what you're saying about the cystectomies?
So you can do the cystectomy where you take out the ceca, because there's not much microbial fermentation happening within the large intestine or the colon of the bird. It's only, an adult bird is only about 2-3 inches long. So you can just surgically remove those, and then
They
you
don't need
can, oh, it's like us with our appendix. So there's 2 different ways. One is terminal, one is not. I think we'll end here.